Hello everyone, thank you for joining me to this webinar called The Missing Data, Why Translational Insights Are the Future of mRNA Innovation. I'm Arnoldi Michele. I'm Technical Support and Product Specialist here at Immagina. So for who of you that doesn't know who we are a small group of 15 people that work in our Immagina headquarters in Trento, in the north part of Italy. We do have our building of 1,500 square meters, which have both innovation, service, and production, because we do produce our own kit here in our headquarters. But for logistics reason, we do have also Immagina subsidiary Inc. in New York, in the US, so to have an easier and fast shipping in the north part of US, America in particular. And then we do have a wide variety of distributors through all the globe. Before starting my presentation, I would like to tell you that we will have a Q&A section at the end of the webinar. So if you have any question related to any slide, or if you have curiosity, please let me know at the end. You can write on the chat, or you can ask to directly talk. Okay, so we'll start with the first part, which is why RNA is at the center of biotech and pharma innovation. Well, the concept of RNA therapeutics was first discovered in the 60s through the discovery of the mRNA, but the concept itself arose during the 90s. But the actual prominence and why these RNA therapeutics can be very important rise after the mRNA vaccine during the COVID-19 pandemic. So those Pfizer, BioNTech, and Moderna vaccines unequivocally prove that RNA can be safe, they are efficacious, and it can be scalable through all the globe. So nowadays, there is much more importance toward this RNA therapeutic, and in fact, only in the last quarter of 2024, there was a 53 boost of RNA therapy trial initiation, with a market growth that is 13.3 billion in 2023, which is supposed to become 21.43 US dollar billion in 2032. So why is this so popular? Well, there are a few reasons. There are about 1,261 RNA therapies in pipeline, from both preclinical to pre-registration. And the way why it's becoming more and more relevant, this field, is because it's more versatile compared to drug targeting proteins. The reason why is because there is the so-called druggable proteome that defines just a fraction of protein, which have the really way in order to bind through the affinity, the specific target. They need to have properties, and the protein to be targeted are somehow limited. They are supposed to be about 1,500. Though the druggable genome, so-called, so everything that can be druggable with those small molecule and antibody, is a bit bigger, about 3,000, is supposed to be in the future. But there are more diseases which are not easy to target with canonical way. And that's why the versatility of RNA can come into place, because it goes beforehand from where the protein is. Okay, so since there is interest in this topic, also the different agency, I just reported European Medicine Agency, because we are in Europe, wrote guidelines in order to tailor which could be the different aspects in order to create RNA vaccine. These drafts provide guidelines specifically to define what is an active substance. And it can also define what is a sequence that has to be a 5' cap, which are the UTR, poly-A tail, etc. So you do have information regarding RNA quality, structure, and functional feature. But we have to remember that an RNA is lately supposed to be translated into a protein. And those kind of measurement in order to assess what is the efficiency of this passage from RNA to protein is not really detailed, creating a so-called missing gap. This is why the title of our webinar. But going from what we can do in order to change and create better RNA therapies, well, we can engineerize them in order to create better stability of the transcript itself, have a higher functionality, and minimize the immunogenicity. I will just report a few examples. I mean, this is mostly an overview for you to have an idea. We can go more into detail, but the overall goal is that you can design and optimize, for instance, the 5' UTR. And here you have 3 different type of structure that have been engineered. Those structure can create different structure motif that can hamper the initiation phase or can facilitate the initiation phase. So you can play how the translation work also starting from the 5' UTR. But lately, what you can also do is to select which are the optimal codon in order to influence translation efficiency, because have been proved that if you change the codons, not changing the protein itself that have been produced at the end, but changing the codon, you can let the ribosome move faster or slow it down. And these have been linked also to the transcript half-life, because you can prolong the half-life of transcript or shorten it down, depending on which are your goal. And these impact, so the ribosome translocation, the half-life, and overall protein production. But we have to remember that the RNA can also form secondary structures that might hamper the movement of the ribosome itself, because you have to remember that when the transcript is translated, the ribosome needs to move on a single strand RNA. So those local text structure might decrease the kinetic, and so you need to also take into consideration which is the global secondary structure in order not only to promote or slow down ribosome passage, but also to change the half-life again about the transcript itself. So this is just a schematic to basically summarize what I just told you, because what you need to do from an original RNA is just to find out how to boost the half-life if you want that it lasts longer inside your cells in order to present and be possible to produce protein. Only increasing translation might not provide the same output because you're boosting for a short period of time, but then the RNA is degraded, so you cannot produce anymore. So the idea overall is, if you really want to have a prolonged effect, you might want to play around also to increase both translation and half-life. What we need also to remember is that RNA can be very much modified. And depending on which kind of modification you insert, you can change completely how the RNA first of all, is recognized by the cell. So you can reduce immunogenicity, for instance, but you can enhance stability and expression. So you can enhance translation, you can decrease the stability, and so on. So remember that also those aspects need to be really taken into consideration when designing an RNA therapeutic. Okay, so overall, what I just reported to you is that in order to really understand the expression level of the target protein from your mRNA sequence, you need to take into consideration the different UTRs, the codon usage, the secondary structure, and the modification, in order overall to play with the ribosome dynamics and how ribosome move, if they are too fast, too slow, if the RNA becomes unstable, and so on. But what is missing also is that in order to produce a protein, we need other aspects. And another player that play a pivotal role in translation is the tRNA. So not only you need to really check your RNA of interest, but you also need to check in your in the cell in which you want to deliver this kind of RNA, which is the tRNA abundance, and if they are correctly modified, because maybe the cell of interest do not have the correct tRNA, and the protein production is difficult. tRNA are some of the most modified molecules, and they need to have specific modification in order to elicit their role. Another aspect to consider is that when you have an RNA sequence which is supposed to translate, you have a canonical translation. But somehow there are a lot of reports that suggest that you can also have alternative translation from a critic ORF. So you might have upstream ORF, downstream ORF, and all of those might produce some byproduct that can be very harmful if you think about an RNA therapeutic. So also knowing if there are other proteins, small protein in this case, being produced might be important. So simply knowing the RNA structure might be insufficient, and you need to think about understanding also how to optimize this other aspect in order to achieve the better or the desired protein expression. So for this, we need to have advanced technologies such as RiboSeq and tRNA-seq in order to really dig into those kind of events. So the second part is, okay, but how do we measure those events? Well, in general, in order to measure those events, we need to do a step back. First of all, if you have your RNA, mRNA in this case, in order to be translated, it needs the ribosome, which with auxiliary factor. Since there are different steps in translation, I did not include any. This is really a schematic. You need the tRNA in order then to produce the protein of interest. So historically, how to measure those, the first way in order to measure what is actively translating have been have become possible thanks to the introduction of the polysome profiling. Now, for who of you which is not familiar, you have a sucrose gradient, and you are pelleting down through ultracentrifugation the different population of ribosomes, so subunit, ribosome, or polysomes. And then you separate them through fractionation, but then you can extract the RNA and do RNA-seq, for instance. Another possibility is to provide a tag to the ribosomal protein and then have an antibody in order to fish out the ribosome, then extract the RNA underneath, produce library and sequence. In 2009, ribosome profiling came into place, which utilized a similar technology, so still ultracentrifugation and sucrose gradient, but provide spatial information, because what you can know is where the ribosome was set. in this experiment, you utilize a nuclease in order to cut all the RNA but the one protected by the ribosome. In this way, you create a ribosome protected fragment. Those little fragments were protected by the ribosome. And by collecting those and doing library and sequencing, you know exactly where the ribosome was sit, hence which part of the transcript was translated. Lately, what you can just do is to palette or all the ribosomal complexes which are translating, and then do the RNAs. So starting with this global scenario of what was the correct status, we developed a new technologies called ribolase. Instead of using the bulk equipment such as the ultracentrifuge and fractionation, we came out with an idea of using magnetic beads. So everyone can use this technology on their bench. How does it work? Well, what you have is a cell or tissue, it doesn't matter. You do lyse. Then we do provide a nuclease in order, similarly to the ribosome profiling, to cut all the RNA but the one protected by the ribosome in order to ease the complex structure and have so-called such as individual ribosome with the ribosome protected fragment. We do have a probe called 3P, which is an analog of pyromycin. This analog is able to recognize actively translating ribosome thanks to their conformation. Since this probe is linked to magnetic beads, what you have to do is just to use a magnet and fish out all the actively translating ribosome. Then you do extract RNA, and we couple this ribolase technology with an ad hoc library preparation called LaseSeq. So this library takes advantage of the 3' phosphate signature, which is left there by the nucleus digested. Since only RNA which have been digested contain this 3' phosphate, and the majority of the short RNA will be ribosome protected fragment. So those little fragments are inserted specifically into our library that then can be sequenced on Illumina platform and then analyzed. The good thing is that there is no gel extraction because everything is without any gel, which is different from the canonical ribosome profiling, which require gel extraction, both to select the RPF and to clean up the library. We do not need that. And thanks to that, the workflow becomes 2 to 3 days. This is very compatible with a lot of different cell and tissue. So far is for eukaryotic specimens, specifically mammalian, but it can be expanded to other. This is regarding the ribosome profiling. But if you remember, in the beginning, I told you that another aspect that is important to consider is the tRNA. So we also acquire a technology called nanotRNA-seq, which enable you to study the tRNA content in the native form. How does it work? So basically, you do need to extract total RNA from your species. Here you do not have any limit. The only limit is provided by the fact that you need to have the tRNA sequence, because then you need to align a sequence to a reference. So if you have a very weird archaea, it's fine because you can extract total RNA with whichever kit you like. You just need to have the tRNA sequence annotated. Then you extract the small RNA because the RNA are small and they are present in the small RNA part. You deacylate in order to remove the amino acid loaded. Then we do have a splint adapter which is able to recognize the clove leaf shape of the tRNA. Then we do have adapter ligation in order for you to multiplex up to 6 samples in one library. This streamlines the sequencing because it enables you to sequence in one flow cell up to 6 samples, because we do have a way to demultiplex this and have individual libraries. These we then combine the first ribose and this nanotRNA-seq in order to create a new kit to study the sequencing translational active tRNA. Because we realized that when you pull down the actively translating ribosome, you're not only pulling down the ribosome protected fragment that is originated by digestion, you're also pulling down the tRNA which was involved in translation. So using the same library as I just mentioned to you, you can sequence through Oxford Nanopore the actual tRNA that was undergoing translation. The good thing about both the nanotRNA-seq and the so-called ribotRNA-seq kit is that using Oxford Nanopore platform allow you to sequence native RNA because it's a direct RNA sequencing. So you can have real quantitative data of how many RNA are present, plus you have all the modification information, because the RNA are some of the most modified molecules most modified RNA in a cell. So you can also have information of which kind of modification you have. So for the last part, the idea is to show some real world case study in order to really understand why it's important to really play and optimize your RNA for therapy. The first thing that you can study is how many ribosomes are loaded on your transcript of interest. And this group, what they did was to perform polysome profiling on different GFP. So they took exactly the same GFP, producing the same protein, but they changed the codon usage. Then they did polysome profiling, and they count how many ribosomes were loaded on every transcript. And what they saw is that depending on how you design your sequence, depending on which codon you use, you can have between 5.5 to 8.5 ribosomes loaded on them. Interestingly, the amount of ribosome was inversely correlated with the half-life of this mRNA, having the one with 5.5 ribosome loaded on them the highest half-life. This was also coupled with more protein production calculated by total fluorescence synthesis EGFP, and this is a way that I want to pass this information, that it's really important to really design your sequence with the correct codon in order to boost translation, but also to let your RNA to be stable for a long time. In this way, you can maximize the output, because in this example, the highest level of protein output was done in the sequence with the highest half-life. Another real-world case study that I want to share with you is the human coagulation factor 9 fix of the hemophilia B deficiency, which is a focus for gene therapy clinical trial. So what they did was to take the wild-type fix sequence and then create a codon-optimized fixed and compare them to ribose-seq. So they perform ribose-seq, and here you can see the cumulative sum of how many reads you have in that specific position thanks to the ribose-seq experiment. What they did then is to sum up all the signal and normalize it. And what they found is that by optimizing the codon here in red, you do have more reads compared to the wild-type form, which is not the case in two controls such as actin B and GAPDH. So somehow ribosomes are more present in the codon optimized fix compared to the wild type form. But when they looked at how much these two proteins were produced, they did not see changes in translation efficiency. So somehow these translational kinetic do not resemble in increased protein production. What they found is, however, that the different kinetic might affect the protein folding, somehow telling you that you have also to consider the very last part of protein production, so folding of protein, because in order to elicit the role, they need to be properly folded. So somehow here ribosomes have a different kinetic to promote, possibly, the protein folding. Another paper that I want to share with you is this one, in which they utilize actually our ribosome kit to perform ribosome profiling in cell in cell infected with HIV or not infected as control. All our kit and our services are certified because we have the quality standard. So what they found was that in the UTR region of the HIV genes, they found a non-AUG translation initiation site, and this was not seen before, and is proving another time that it's important to know whether you have byproduct of your RNA sequence, because it might be that translations start from unexpected regions. Okay, but all the three examples that I share with you are mostly related to how to see the ribosome dynamics, possibly through RiboSeq. What I want for last to share with you is an example of what you can study on with the tRNA level. This is a manuscript that we are preparing, so stay in touch for when it will be released. This is just an example. So what we did was to treat cells with nothing or with arsenate for an hour. These hamper the translation, which is proven here by polysome profiling with a reduced polysomal fraction. What we did was to extract total RNA from those cells or perform ribolase. So we pull down actively translating ribosome with our probe. We then did tRNA-seq of the two population to have the total tRNA-seq, so all the tRNA present in the cell, or the ribo-tRNA-seq, so only the tRNA within the actively translating ribosome. When we look at the total tRNA content, basically no changes were observed in arsenate compared to control. But when we look at specifically the ribo-tRNA-seq, we observe quite a lot of differences, both up and down regulated tRNA, supporting the notion that this oxidative stress causes specific changes in the tRNA at the ribosome level. So somehow the cell do not have the time to really change completely the amount of tRNA, but is somehow selecting different tRNA in order to produce the protein needed to reply to the stress. So as a final remark, I just want to leave you with some question. So why tRNA dynamics, ribosome engagement, and RNA structure should be considered during therapy development? That integrating translation analysis, it's very important for any RNA pipeline. That using our nanotRNA-seq and ribolase, this can help you in reducing trial and error in the RNA construct design. And the two questions that I want you to leave with are, do you really know which of your constructs are truly being translated? And are you optimizing the RNA expression or just transferring the transcription? With this, I want to tell you that what I show you today is mostly ribose and lysine and the nanotRNA-seq. But we do have other products, and I invite you to have a look at our website, especially that we do have a workshop that it will be held here in our headquarters between the 25th to the 28th of August. So if you're interested in doing some ribosome experiment in the multiplex version, let us know. So thank you, and I will happy to have questions. See a question from Michele Arnoldi. I have a question: how to ensure that your tool on the ribosome sequencing? So the question is how to ensure that you pull down active ribosomes, particularly in the case of mRNA sequencing, because this is relevant if one is studying the translatome under translational inhibition, since there are mRNA trapped with the 40S subunit. Okay, so thank you for the question. We do know that we are capturing active ribosomes because what I did not include is what you can then have as output from the ribosome. Specifically, you can check the meta profile, so how those reads are aligned on the different mRNA, and you see clearly 3 nucleotide periodicity. So it's like the ribosome are moving 3 nucleotides at a time, resembling what is the triplet that has to be moved. So if you only have the 40S there, first of all, our probe cannot bind because the ribosome need to be completely formed in order for the puromycin analog to bind. Otherwise, there is no binding. Second, it has to have a certain conformation in order to enter and bind. And third, you know that you're taking actively translating ribosome because they are not randomly distributed. They are moving 3 nucleotides at a time on the different mRNAs. Because knowing the positional information is very relevant in this case, because you do know that those ribosomes were actually on the correct frame, and you can have information on the different frames. So frame 0 should have a higher percentage, etc. I hope I replied to your question. Okay, thank you. Okay, so if there are no other questions, I'll invite you to have a look at our website in general and to see our latest updates. And if you have other question or information about product services, please let us know. You can write directly to me or to info@immaginabiotech.com. In this way, we can redirect to the correct people to reply. So thank you for joining me today, and I wish you a good day.